Welcome back, everyone. This video is going to help you understand the parts of the cell and how things are moved into and out of the cell. Part 1, the cell and its parts. This is certainly the most important image in this chapter because it really gives us a map and somewhat of a three-dimensional perspective of these busy places we call cells. Cells are really tiny if you've never had the opportunity to look at them under a microscope. Or if you have, you know that you really have to increase the magnification to see these cells. Cells come in all different shapes and sizes. Of course, we have neurons, which are long and spindly cells. We have muscle cells, which have the capability of shortening. Skin cells are kind of flaky. Bone cells are kind of chalky. And this picture gives us sort of the vanilla generic cell. Okay, so what do we see? Major portions. We start with the cell membrane. That's this blue bubble-looking structure on the outside. I really like how the illustrators show it puckered and uneven. It's as if it's moving, it's jiggling. You'll see in some videos in a little bit that the plasma membrane is remarkably flexible and very thin relative to the size of the cell. The plasma membrane is probably something like the thickness of a balloon relative to the volume of it. What other parts do we see? The other major portion is here, the nucleus. We use this term, of course, to talk about the center of an atom. We also use it to talk about the center of a cell. And you know that the nucleus contains the genetic material, the DNA, the blueprint for life. Do you know what this thing is, though? The nucleolus or nucleolus? That's a big raw pile of RNA, not DNA, but its close chemical cousin, RNA, which is going to be put together into the protein assembly machine, which we'll talk about in part 3 of this video lecture. Okay, so the nucleus is wrapped in a cell membrane, very much like this one. It's a phospholipid bilayer called the nuclear envelope. The plasma membrane, the external cell membrane, is also a phospholipid bilayer. That's going to become very important. So are these things here, these so-called membranous organelles on the inside, the Golgi apparatus. Excuse me. In other classes, you may have learned this as the Golgi organ or the Golgi complex. I don't care. It's the Golgi. You can call it Golgi apparatus for the purposes of this class. This thing here, smooth endoplasmic reticulum. It's kind of a mouthful. And then these little dots here are ribosomes. Ribosomes are protein synthesis machines that are actually made of that RNA we had talked about before. When the ribosomes are on the surface of this membrane, it's no longer smooth, it's now rough endoplasmic reticulum. So let's look back and forth between those. The smooth endoplasmic reticulum tends to be a little bit further away from the nucleus. The rough endoplasmic reticulum tends to be very near to the nucleus. But obviously the most significant structural difference is the presence of ribosomes. Rough has ribosomes. Smooth, no ribosomes. Do you remember what ribosomes make? Protein. So what do you think the rough endoplasmic reticulum makes? Tons of protein. If the ribosome is a protein sewing machine, then the rough endoplasmic reticulum must be a giant fabric factory, right? It's churning out tons and tons of proteins. Anything that's synthesized by either the rough or smooth ER is packaged and walked over to this organelle right here, the Golgi apparatus, which is the shipping department. So look at this. Phospholipid bilayer allows proteins to be packaged into tiny vesicles, which are phospholipid bilayers. They merge with another phospholipid bilayer in the Golgi, and then it bubbles off a phospholipid bubble called a transport or secretory vesicle, which then merges with the phospholipid bilayer of the plasma membrane, and it squirts out the product. This is secretion. Secretion like perspiration and salivation and milk production. Secretion like enzyme production in your gut or stomach, or mucus production in your mouth, saliva, neurotransmitter, hormone secretions. It turns out that the vast majority of chemicals that are produced by our cells in the form of hormones and secretions are sort of spat out of the cell through this process. And the reason that it's so efficient is that they're all made of that same stuff. So one bubble can merge with and become another bubble and then leave the cell. We see a very similar process over here. Lysosomes are these so-called intracellular inclusions. English translation, they're bubbles. They are very much like the secretory vesicles. They are also a phospholipid bilayer. They are a bit larger, and the lysosomes are full of cleaning agents. Lysol, right? Specifically, they're full of hydrolytic enzymes, such that when the cell encounters germs or viruses, it can drink those in, fuse them with the cleaning products, and then spit the junk out, thereby detoxifying and having some sort of primitive immunological protection against the cell. The centriole is this tiny little thing that looks like a pasta noodle. It is always oriented at roughly 90 degrees with another centriole, and so the two of them at somewhat of a perpendicular angle form this structure, the centrosome. This is the puppet master. The puppet master pulls on all of these little strings, these fibrils and tendons and microtubules that we call the cytoskeleton. So here's the puppet master thing. You remember a marionette is a puppet suspended by strings, and the control mechanism is two little wood slats at 90 degrees, making a little cross, right? And you tip it around and it makes the monkey dance. So this is how I think of the centrosome. It manipulates the cytoskeleton and can actually move the whole cell by tugging on the cell membrane or pushing on the cell membrane. It can help control some sort of an amoeboid movement. We'll also see that the centrosome is critical to this process of cellular division that we call mitosis. We're going to divide and chop up the DNA into these little X shapes called chromosomes, and then the centrosomes will pull and push those and ultimately divide the nucleus into two, and then ultimately divide the cell into two. You might remember the cellular division is the way that we grow and heal. Peroxisomes are also little intracellular inclusions, little vesicles, if you will, but they are full of peroxides that help to reduce free radical formation. So antioxidant peroxisomes. It's kind of a mouthful, but that's what you should think about. The cell, of course, is fragile and vulnerable to oxidation, which could cause structural damage to the cell or genetic damage, leading to cell death or a mutation or cancer. The peroxisomes help to reduce the risk of that oxidation by sucking up those free radicals. These little wiggly bits are called mitochondria, and they are arguably the strangest inhabitant of your cell. Although we classify them as an organelle, they are technically tiny creatures in their own right that just happen to live inside of our cells in a symbiotic relationship. How about that? How do I know this crazy, preposterous thing? Mitochondria divide at a different time than the rest of the cell does. Mitochondria have their own DNA and pass it to their offspring in a different way than we do. And there is evidence that mitochondria have lived in primitive cells for hundreds of millions of years, billions of years. We can find fossil records of mitochondria going way back. So this is not a human thing. This is a life thing. Most so-called eukaryotic cells use mitochondria to generate their energy. So here's what you really need to know about the mitochondria. They recharge a tiny chemical called ATP, which is used as a little rechargeable battery to motorize all of these other organelles. Got to move the Golgi, you need ATP. ATP is the battery that powers the Golgi. ATP is the battery that powers protein synthesis and the cytoskeleton movement and exocytosis and vesicular transport. ATP is the universal chemical energy of the cell. The mitochondrion, or mitochondria, recharge the battery. These are the recharging stations. In the presence of oxygen and fuel, the chemical energy from your food is transmitted. No, that's not the right word. It is converted into chemical energy in ATP, which powers your cell. What happens if you hold your breath? You run out of oxygen. And what happens to your ATP? You run out of it. And then what? You grind to a halt. You could even die. If you run out of oxygen, you run out of energy. And without energy, no factory can work. And certainly no cell can work. Okay, wrapping up this very long description of this one picture, there are a few plasma membrane modifications that you need to know. What the heck does that mean? Well, the plasma membrane is the cell membrane, and it's usually sort of fluid and smooth like a bubble. But sometimes it has these long, wiggly, hair-like structures on it, and sometimes it has these short, fuzzy, hair-like structures on it. The short, fuzzy ones are called microvilli, and they don't tend to move, at least actively. They might get pushed around or wiggled by fluid moving past the cell, but microvilli they're not motorized. Microvilli are there for increased surface area, for absorption or secretion. The cilia are motorized. They do wiggle very quickly, maybe even a couple thousand wiggles every second. Humming and buzzing right along, the cilia help to move fluid along past the cell. In this way, our body can actually generate fluid currents in such places as the fallopian tubes and the respiratory tract to help to flush things along and sometimes to clean or debris the regions of the body. All right, moving on. I'm not going to read the entire PowerPoint to you, but you can pause this periodically to take a look at these important bullet points that I have laid out for you. Functions of the cell. This really talks about the difference between living things and non-living things. You are alive. You can use energy in a process called metabolism. Now, metabolism really speaks to the ability of your body to harvest chemicals and the chemical energy within your food, and then to use those chemicals and that energy to build your body out of the food that you have eaten. Whoa, that's crazy, isn't it? You are what you eat, both physically and energetically. Living things also have the ability, as a part of metabolism, to synthesize new things. They can take one chemical and another chemical and merge them together into something bigger and something more complicated, something that has more energy and maybe even more intelligence in the form of storing chemical patterns like what we see in DNA, RNA, and proteins. The third bullet point, communication. Cells have an amazing ability to communicate with their neighbors, to tell them if they're feasting or starving, if they have nutrients to share, or if they've received a chemical signal, or if they're dying. These chemical and electrical signals really allow a small part of the body to communicate with the body as a whole. So part of my job this semester is to really blow your mind and help you understand that you are not really a single organism. You are not a fish, people. You are a coral reef. Okay, yeah, you're a big mammal and you have mass, but here's the thing. Not only are your cells communicating, but the viruses in your body and the trillions of bacteria, protozoa, and fungi that colonize your body are also in communication with your body. This sounds sort of strange and out there and metaphysical, but it's not. I'm talking about actual physical symbiotic relationships that work in ways like this. The things that you eat affect the ways that you think. The things that you breathe in can affect your metabolism. And obviously, we know that diet and nutrition and the potential drinking in of synthetic chemicals can have serious effects on our cells' ability to communicate. And we might even be seeing that in strange ways in the prevalence of type 2 diabetes, in neurodegenerative disorders or neurodevelopmental disorders. Lastly, non-living things cannot make more of themselves by passing on their characteristics, right? A rock doesn't, well, maybe it does. A rock breaks apart into more rocks like itself. Oh my gosh, that's crazy. All right. In all seriousness, living things can reproduce, right? Either asexually, like sponges or bacteria, which sort of clone themselves, or, as mammals do, a much better way, in my opinion, sexual reproduction. Not only is it enjoyable, but it's really good science, right? It's a great way of passing on your genes, because if you survived long enough and did a good enough job of attracting a mate, then nature says, hey, you deserve to pass on your genetic characteristics. And we call that inheritance, right? So it's not an accident that I look like my grandfather. And actually, if you could go back 7 or 8 generations in my family, you might even see that my great whatever grandfather, that I bear resemblance to him as well, right? Survival of the fittest. If they survived, then they got to pass on their genes. And so we are all survivors, which is pretty amazing to think about some of the struggles that our ancestors may have gone through. And your physical body is a legacy of your ancestors' ability to survive and to attract a mate. So maybe they were really good dancers or singers or romantic or strong, or they were really good at gathering food, or they were really good at protecting their farm. Whatever it was, you were meant to be here. Some cells are specialized for this exchange during sexual intercourse. We'll talk all about that in the next semester, 202, sperm and egg, and what happens if you get them too close together. Okay, man, there's so much detail in this chapter, but we can just dive right in, looking here at the plasma membrane in a static perspective. Hopefully you've already had a chance to watch some of the YouTube videos to really see how fluid it is. But we can see here the most important concept is this. You are what you eat. What? What do I eat? I eat carbs, fats, and proteins. It's right there on the label of my little candy bar. You can see it clearly. And here too, on this figure, on slide number 7 of chapter 3, what do we see? Carbs, fats, and proteins. The carbohydrates are these long chains. This looks like glucose, 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 fructose, glucose, glucose, right? A long chain of simple sugars. Not that dissimilar to a croissant, actually. Lipids. Primarily we see these phospholipids. I'll talk a lot more about them in just a little bit. The phospholipids and cholesterol are the two most important lipids of the cell membrane, and they balance out this ability to be flexible and not too fragile to rupture. So as long as you have enough cholesterol here, the phospholipid will stick together. If you take too much cholesterol out, the cell becomes too fragile and it begins to break apart, which is a bit interesting if you know anything about statin drugs and their most serious complication, rhabdomyolysis, which is exactly what I described right there. Proteins. When it comes to proteins, people, it's all about the shape. Look at this. This one's shaped like a tube. This one's shaped like a keyhole. This one's shaped like a scissors, right? You might have to use your imagination, and the illustrators have used color to really help draw our eye to these different shapes. But that's what we're really going to understand in part 3, if I ever get to that part of this video. The DNA is going to determine, ultimately, the shape of the protein. And like everything else in this class, the structure of a protein is related to its function. So look at this. It's shaped like a tube. What do you think it does? It's a channel. It's a doorway. It's a tunnel. This one's shaped like a keyhole. It receives chemical signals, which work like little keys to turn the cell on or turn it off. These ones might be shaped like scissors, cutting things apart like an enzyme. Here we can really appreciate the complicated three-dimensional structure of the protein. And we'll find out in even more detail how that structure will determine the function. Look at this. Keyhole, scissors, tunnel. This one's a door that can be opened or closed. A gated ion channel may be opened or closed by touching it, or maybe by zapping it with electricity or stimulating it with a hormone. These become really important when we move into neurology and the endocrine system. Cell identity markers are also known as antigens. So these are used by the immune system and used by one cell when it bumps into its neighbor to know, hey, is that a good guy or am I bumping up against an infection? Germs have different cell identity markers, and so our immune system uses these ID tags to tell friend from foe and to tell good cells from bad. Cell adhesion molecules have a unique shape which allow them to stick together a bit like Legos, giving a very strong connection between one cell and another. Examples like that include dense connective tissues, our skin and bone. Okay, we're going to be moving into the basics of transport in just a little bit, but we really have to see how these transport proteins' shape can allow them to do different things. Look at this. Some of them work like channels, just allowing a substance to flow from one side to the other. Some of them are going to work more like a turnstile. Do you know what that is? When you go to the movies or you go to the Brewer game, you have to walk through that little three-armed thing. Or at the subway. In the tube. We're going to see pictures of that in just a second, how those proteins bind with the substrate, and then they change their shape themselves simply by binding to that other chemical, and then move it through, and then the protein flops back to the other side where it can pick up another one. So it's sort of like the turnstile at the security gate. You got to go through. This thing is not motorized. It's not using energy. It is simply changing its shape when it weakly binds to the transported molecule. Maybe the poster child for this one is glucose. Glucose can't get through the plasma membrane, but has to rather go through one of these so-called transport proteins. ATP-powered pumps are another very important way of moving things through, and we can see an example of it right here. Ions also can't go through the plasma membrane but have to be moved through. And look what happens. When the ion binds to the transporter, energy is supplied by the little battery, ATP. And when we break that chemical bond, we no longer have 1, 2, 3 phosphates, adenosine triphosphate, but we have only 2, adenosine diphosphate. And the breakdown of that releases energy, causing this to work like a little pump, like a little squirt gun. It pushes the ion out. And in this way, we're using energy to move something in a pumping motion. What a great name, active transport pump. Enzymes are also proteins. When it comes to proteins, it's all about the shape. The shape of this enzyme is such that it will only bind to this chemical. And when it does, it snips this apart, releasing the individual components. This is a decomposition reaction called hydrolysis, and it wouldn't happen, or certainly not fast enough, were it not for this enzyme. Keep in mind some enzymes work in the other direction, building smaller parts into bigger ones. So you have to imagine this is a double-headed arrow, although each enzyme would only work in one specific direction. We have two enzymes, one that puts them together and one that takes them apart, as you see here. Great summary of what we just talked about. You should make a flashcard for each of these and make sure you have the function on the back side, and then review them and make sure you know what all of these things mean. One of the last parts of our discussion here in part A, holy cow, this is a long section. One of the last parts we're going to talk about in this part A video is the cytoplasm. It's like the jello of the inside of the cell. It's made of fluid, intracellular fluid, or cytosol. And then all the other stuff we talked about. The organelles, like the Golgi and the rough ER. The cytoskeleton, like the microtubules and fibers. And those cytoplasmic inclusions, vesicles, lysosomes, peroxisomes, all of that together is the cytoplasm. And when we come back in video B, I'm going to pick it up here by going into a bit more detail with the cytoskeleton. Go take a break. You deserve it.